All right, so more about replication part two, and we're still talking about DNA replicating itself. And we discussed in the previous video that DNA basically can be split into the two ribbons, and these ribbons or these strands can be replicated or cloned basically into a daughter strand. And we get this original strand and this daughter strand that forms, and it's the same DNA that we ended up or that we started with at least. And we said that there's certain things that go on within this whole description that allows the DNA to be separated, that allows the DNA to be replicated, and then basically glued back together in a way. So that's what we're going to continue on with here. We're going to keep talking about how DNA replicates itself and what are some of the requirements and more terminology that we use to describe how this thing repeats. So first off, up at the top, you're going to see replication requires a separation of the double helix. This is something that we've mentioned before in the previous video. So this DNA strand that is very stable, that is glued together by hydrogen bonds, that has this corkscrew turn on it, and that has all of these polar regions on the outside of the backbone. This thing has to be ripped apart. And if it can't be ripped apart, then we can't replicate it. That's the whole point of this replication, is that we're using the original DNA strand and we're going to have to replicate the original somehow. So that's the first thing that has to be done, is that this thing has to go in and basically unzip itself. So we can't really do that. We can't allow DNA to unzip itself on its own. But our bodies can create something that we call a helicase. And the helicase is basically a protein. That's all that it is, a protein made up of an amino acid chain. And the helicase will basically be like your finger. And the finger will go onto the DNA and grab a hold of that zipper, and it will begin to unzip the DNA into the two strands. And that's the purpose of this thing called the helicase. Now, the helicase has to have some type of energy, and that energy is going to come from ATP. And this ATP we have talked about before, because this is when we talked about the adenosine, and the T stands for tri, and the P stands for phosphate. So this is an energy carrier or an energy source. And up here at the top of your PowerPoint, you can't see it on the screen, but you're going to see the structure of ATP here. So this is very important. This is an energy reserve system that helicase uses in order to do its job. And that job is to unzip the DNA into the two strands. So you're going to see the adenosine up here at the top, and then you're going to see the ribose sugar that's here, and then you're going to see 1-phosphate, 2-phosphate, 3-phosphate, so hence triphosphate. So the helicases can act what we call passively or actively. So that basically means they can be there doing the job, or they can be there not doing the job. And the new DNA strands are always copied in the 5' to 3' region. That's something that we mentioned previously, and that's something that I mentioned again because it's so important to talk about that, because this does give rise to other things and other components and other players in the game that some people do leave out. Okay, so let's take a look at maybe a better picture of what's going on here, and then we'll come back to this slide. This is the helicase, and the reason that I like to show this picture of the helicase is to let you know that the helicase is as complicated as maybe the DNA is in itself. And that's because we have these primary amino acid sequences that have came together. The amino acids have basically created secondary structures, which are alpha helixes and beta pleated sheets, and you can see those in the structure. The alpha helixes are these corkscrews or these curls, and the beta pleated sheets are these purple flat areas that are here in the center. And then the secondary structure has created a tertiary structure, which is how do all of these things basically give it some shape. And then a quaternary structure is really the computerized 3D model of all of these components and how they hook up together. You can kind of see there's 4 fingers if you want to think of them that way. There's a finger here, one there, and one here. So 4 4 fingers that latch down onto the DNA and begin to unzip this thing and open it up into the 2 strands. Okay, so that's the picture of the helicase. Again, just a protein, that's all that it is. It gave me a chance to go back and talk a little bit about that protein chemistry and amino acid chemistry as well. Here is the structure of what we want to look at as far as how this thing begins to separate. So if you go through and try to find the picture in your slide, you're going to see the original starts up here at the top. So here is the original. And we see a 5' end here, and then that will curve around and around, coil around, and we see the 3' end on this side. And then here's the 3' end with a complementary original strand. And if we corkscrew that around and around, we can end up with the 5' end on there. So those strands run anti-parallel. Those are the original DNA strands. We said that one runs 5' to 3', the other one goes from 3' to 5', so they run in opposite directions. So when we begin to separate this, so that's what you're seeing here at this location. This thing is beginning to get separated. We see one of the parent strands, and that's in the dark blue here, and we see another one of the parent strands that's in dark blue there. And then we start to see the replication come in and take place. So the separation happened because of the helicase. The helicase goes in, it's a protein, it's an enzyme. It uses ATP to get enough energy to start ripping apart these hydrogen bonds and separating them so that they can start to get replicated. And what we notice is that on this end, this is the parent strand, and that's the 3' end here. So we start replicating that. So the parent strand has an A, the complementary strand would bring a T in. The parent strand has a G, the complementary strand would bring a C in. And then that goes all the way up through the parent chain, right? So here's the parent chain of G. The daughter strand would bring the C in. And then there's a C. The daughter strand would bring the G. And what we're doing is that we are actually cloning this DNA strand, because once this goes all the way through and replicates the entirety of the actual structure, we actually see that this strand is no different than the other strand. They are identical copies of each other. The only difference is that one of these strands is the original and one of these is the copied one. So if we take a look at this parent strand at this 3' end, this is very important because that means that this daughter strand that's getting replicated, or that's becoming replicated, that is being synthesized in the 5' order, and that's the proper order that this thing should be replicated in. So the 3' end on the parent strand starts simplifying and synthesizing this 5' end of the daughter strand. So the 5' end goes up and it continuously becomes replicated over and over and over through the chain as this DNA strand becomes more and more separated. So this is a continuous synthesis on the left-hand side. So it keeps going and going and going and going and going. Now the problem is that on this end, here is the parent strand, and this is the 5' end. And on the 5' end, we actually see the 3' end getting replicated. And we've said that the 3' end, well, it doesn't replicate that way. It has to start at the 5' end and then continues on to the 3' end. That's the proper order that it replicates. So this has to replicate backward. And that's the issue there, because the DNA doesn't want to replicate backwards. So what it has to do is that it has to open up a little bit and start up here at the top. And then it's got to go, okay, well, here's the 5' end. So then I'm going to go through and I'm going to go, replicate, 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 replicate. And then I get down here to the bottom. And then what has happened is that this DNA strand in the meantime has separated even more on up through here. And the strand has to go back, and it has to start at a new location and replicate, 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 replicate until it gets down to the point at which it started. And it's got to continuously do these fragments over and over and over and over. It's not a continuous replication process. Now, this one over this side, well, one of these strands, well, 5' to 3', if it's being replicated that way, it's continuous. That means it starts off at 5' and it continuously replicates that strand as it goes through without an issue. But on the other side, you're going to see one of these strands being replicated in fragments. And then in these fragments, we call these Okazaki fragments. And that's what you're seeing here on the end of this ATP slide that we looked at a while ago. So down here at the bottom, we've talked about the synthesized and 5' to 3' region. One strand is continuously synthesized from 5' to 3' without a problem. The other strand has to be made in pieces, in chunks, and these chunks or fragments are called Okazaki fragments, and this is what we can call a lagging strand. So what has to happen is that these fragments are kind of replicated in groups at a time. So one fragment is replicated, and then another fragment is replicated, and another fragment is replicated, and then the DNA has to make sure that all of those fragments kind of make one continuous message, and we have to join those fragments together. And when we join those fragments together, that is joined by something called a DNA ligase. So the DNA ligase is just kind of going through and kind of looking at the strand as it gets replicated and says, okay, well, here's a fragment and here's a fragment. I just want to make sure these fragments are hooked up together now. So it creates that linkage that connects the fragments together, the Okazaki fragments together. So Okazaki fragments are happening because of the way that the DNA has to be replicated, and then the fragments are finally glued together with the use of what we call a DNA ligase. So very important, again, terminology-wise, definition-wise, that's really what we're after here. That's what this lecture is really all about. The DNA chain is replicated using a DNA polymerase as well. So what happens is that human DNA polymerases, well, these are proteins again, 900 to 1,000 amino acids long, and these polymerases are basically adding the nucleotides to the strands. So this polymerase is basically bringing in the ATs, Gs, and Cs and connecting them together. No known DNA polymerase can begin a new chain. So we often talk about primers. Primers are very important in DNA replication. We have to bring a primer into the mix, and the primers basically are a couple of bases that start off the replication process. So again, more definitions, more terminology, more proteins that do the job of replicating